So here we're going to understand specific activity of an enzyme, and we're going to do this by following along an example, and then we're going to come back and look at these definition terms. So within the cell, as an example, let's take a look at this reaction where pyruvate is converted into acetyl-CoA. So pyruvate is that final product from glycolysis, and it's converted into acetyl-CoA so that it can be fed into the citric acid cycle, where it's going to help to produce these reduced electron carriers that are going to go on to the electron transport chain to make ATP. Now this reaction, it's catalyzed by pyruvate dehydrogenase, and TPP, lipoate, and FAD serve as cofactors that facilitate this reaction, along with NAD, which is going to get reduced into NADH. Now, let's say that as biochemists, we want to isolate this enzyme because we want to study the structure and function of pyruvate dehydrogenase. How would we go about doing that? Because within the cell, we have a lot of different biomolecules, and we want to only purify this enzyme. So in that case, we're going to lyse the cell, we're going to expose all those internal biomolecules, and we're going to follow a purification protocol to hopefully be able to isolate pyruvate dehydrogenase. Now let's say that we have followed our purification protocol, and now we want to test whether or not we were able to isolate pyruvate dehydrogenase. And in order to test if our protocol has actually worked, we can perform an enzymatic assay. And an enzymatic assay will detect any enzyme activity within our sample. So what we're going to do is we're going to take a sample of our purified pyruvate dehydrogenase after following our purification protocol, and we're going to combine it with pyruvate and NAD, and we're going to utilize a buffer, add this to a cuvette, and then measure any change in absorption in a spectrophotometer. Now, how are we going to be able to observe that change in light absorption? What's causing it? So if our sample does in fact have pyruvate dehydrogenase present, and we mix it with pyruvate dehydrogenase should convert the pyruvate into acetyl-CoA, and as a result, NAD+ should be reduced into NADH, and NADH will strongly absorb light at around 340 to 400 nanometers. Now, when it's absorbing light strongly, we're going to see that absorption in our spectrophotometer, and the more pyruvate dehydrogenase we have, the more absorption we will see, because we'll see a greater peak in absorption because there's more NADH being produced. So once we have that enzymatic assay and we have now determined that we for sure have pyruvate dehydrogenase because we've detected enzymatic activity, the next step is to quantify how much protein we started off with and how much protein we ended off with. Because remember, an enzyme is simply a protein, and we want to determine the relation of how much of the protein that we have isolated is actually the enzyme itself. And that is when specific activity comes into play. So once we've performed that enzymatic assay, once we have quantified the protein via one of the quantitation methods, we then determine specific activity, which is a relative understanding of how much enzyme we have in comparison to the total protein. Now, going back up here, now we can look at specific activity definitions, where we can see specific activity is the enzymatic activity divided by the amount of protein that we have. So the units for specific activity are going to be activity units, which could be absorbance per min, it could be, moles per min, whatever the activity is. It's going to be divided by the mass, and often terms that mass is just defined as milligrams. And it tells us, essentially, specific activity tells us how much of the total protein is in fact the purified enzyme itself. And as we move towards increased purity, specific activity will also increase. So in a sample that is completely pure for pyruvate dehydrogenase, that is the maximum specific activity value that we will obtain. So essentially, once again, specific activity is just a relation to how much of the purified sample of protein is in fact the enzyme itself. And in order to do that, we first have to perform an enzymatic assay, and then we quantify the protein, and then using those two values, we determine the specific activity.